All right, team, welcome back to Banana Lab Part 2. For this part of the lab, I've gathered a few more supplies that we didn't need for Part 1. 2 microscope slides and cover slips, my iodine and water, a microscope, toothpicks, and these teeny tiny little test tubes. So in this part of the lab, we're actually going to be able to look under the microscope and see the cells of the banana and the parts of the cells, which are called organelles, that actually contain the starch. The organelles that contain starch are called plastids, and they're going to look black under the microscope because the iodine will have stained them since they contain starch. To see the cells, we only need a tiny little bit of banana. First, I'm going to prepare my iodine solution. I'm going to use this toothpick to scrape up just a tiny bit of the ripe banana. Now I'm going to place that banana into the test tube with my solution of iodine. After I put the banana in, I just shook up the test tube to make sure that all of the banana got mixed with all the iodine. Now it's time to make our microscope slide. Now my slide looks like this. Now it's time to take it to the microscope. When you use a microscope, the first thing you have to do is make sure it's turned on. That will turn the light on. Microscopes have 3 objective lenses that are each a different power of magnification. When you start to focus, you always start on the smallest objective, which is the red one, and it's 4. It's important to know that this eyepiece is also magnifying 10 times. So really, when you use the small objective, which is 4 times, and the eyepiece, which is 10 times, you're looking at something 40 times bigger than it is in real life. Sides of the microscope are focus knobs. The big one will move the stage up and down, and the small one just does fine focusing. Now let's pick up our slide and put it on the stage of the microscope. Now I'll focus it so I can actually see the cells in the plastids. Under the low power, this is what it looks like. So the black dots are the plastids. The procedure says that I should move it to medium power, so that's what I'm going to do now. I refocused the microscope, and this is what we got. Count these plastids. Record whatever number you got in the overripe count 1 section of your chart. Now I'm going to move the slide around so you can get counts 2, 3, and 4. Remember, you're just counting the black circles or ovals that are inside of the cells. Definitely recommend pausing the video to count. Use this for count 2. This is count 3, count 4. Once you've got all your counts, it's okay if you estimated a little. You need to average them. Do that in the last column of your chart. Now I'm going to repeat the same procedure for the green or unripe banana. So now I've set up the green banana and we're going to get our counts of plastids. Remember, the plastids are the black ovals or the shapes that are inside the cells, not the cells themselves. Pause each count so that you can get an accurate or at least a really good estimate. Count 1. Count 2. Count 3. Count 4. I also want to show you what this green one looks like when it's on low power. Don't try to count this one. Here it is. Now all you need to do is average your counts, draw your graph, which is just a graph of the averages. So it's a bar graph. You should have 1 bar for green and 1 bar for ripe. Draw the graph on OneNote and then use the snipping tool or a screenshot to get it into your doc. Then answer the questions underneath the graph and you're good to go. Turn it in. Thanks for learning about bananas with me today.